Yep. My name is Michael Britton, and I am presenting a poster on a comparative study I conducted on two distinct reproductive strategies in ferns, looking at the completely dimorphic royal fern, cinnamon fern, right here, and the hemidimorphic royal fern, right here. And what that means is when it's completely dimorphic is there's sterile fronds, which are completely photosynthetic lamina, whereas the fertile fronds are completely reduced to mostly sporangia. And with the hemidimorphic, the sterile frond again has no sporangia, but the fertile frond has a completely reduced section, but it also has photosynthetic lamina. And in this case, royal fern has the photosynthetic lamina at the bottom and is only completely reduced at the top. What this study attempted to answer was, first, what are the trade-offs involved in dimorphy in ferns? Second, how do these trade-offs vary over time? And how do these trade-offs differ between monomorphic species, hemidimorphic, and completely dimorphic species of ferns? And lastly, how exactly these resources are actually allocated to the fertile fronds in ferns. So one thing we did is we tracked photosynthesis over time, over the course of an entire growing season, and we found some very interesting results. The fertile fronds of cinnamon fern actually died somewhere between day 13 and day 26 of the growing season, and what this species actually did is the photosynthetic rate increased and sort of leveled out until it died at the end of the season. In royal fern, the fertile section of the fertile frond, survived all the way. It died between day 61 and day 75, and in the first part of the growing season, photosynthetic rate increased over the course of the season, but upon the fertile section dying, the photosynthetic rate dropped off, which may tell us that the plant is responding to the resource sink that this fertile frond is. Another experiment we did is we did a cutting experiment. Very early in the season, when these plants were still fiddleheads, we either removed all the fertile fronds or all the sterile fronds, and we had a control where we did not remove any fronds at all. There was no significant difference in the control, which is what we expected, so that's good. When we removed the fertile fronds of these plants, there was no significant difference. And what we're looking at here is the ratio of fertile to sterile fronds. So this is just the relationship of how many fronds of each the plant is producing, and we're looking over at 2 growing seasons. So we cut them at the beginning of 2009 and came back and counted the fronds that were produced in 2010. When we removed the sterile fronds, we got very significant results that the number of fertile fronds compared to sterile fronds was significantly reduced. And that's with the p-value for the cinnamon fern of 0.043 and for royal fern 0.002. And it's somewhat counterintuitive because when you look at completely dimorphic ferns, you would think that they would be much more costly to the parent plant. So upon removing all photosynthetic lamina the prior season, they had no resource gain the prior season, and the totally dimorphic fern still produced a fairly large amount of fertile fronds. You'd figure it would produce less than the hemidimorphic, which would have, originally we thought would have a lower cost fertile frond. And we think a few things come into play here. First thing is that the fertile fronds of cinnamon fern are so short-lived. These things, they mature right here, and then as soon as their spores release, they die. The fertile fronds of this plant probably died within a few days of that picture being taken. Whereas the royal fern fertile fronds survive for a significant portion of the season, and that cost may increase over that season as upkeep and maintenance is required for that survival. And another thing is a difference in the structure of these plants. Cinnamon fern have much larger, much fleshier rhizomes than the royal fern. And that may play a large role in resource storage, so that these plants may have so much resources stored up over past seasons that even though they had no resource gain over 2009, they still had enough resources built up that they could produce fertile fronds the subsequent season. Whereas the smaller rhizomes of Osmunda regalis, royal fern, may not have been able to play that role. And future work that we are going to do on the system is we're going to look at percent carbon, percent nitrogen over the course of the season to see exactly how that relationship is changing over the season. And one thing I'm very excited to do is we're going to do a carbon-13 labeling experiment where with cinnamon fern we are going to label the sterile frond with a bag of labeled CO2 and be able to track that where it goes through the plant. So in one season, we'll be able to see if that carbon is actually moving to the fertile frond in the same season, or if it's only traveling to the rhizome, and we'll be able to see how much of that CO2 is used in the subsequent season. And with the royal fern, we are going to label both the sterile frond and see where that carbon gain is being allocated, and also only the sterile part of the fertile frond, and see if that carbon is either only going to maintenance of the fertile section or if it actually is also going to the rhizome for storage. Thank you.